So good morning, guys. So as you know, you're going to be doing an experiment today, an exciting one where you're going to be extracting some DNA from some fruit. And we're going to first take a tour of the cell and talk about DNA and the genetic code and a bit of what happens in the cell when we process DNA to make eventually proteins that make up your cells and do most of the work or are the workhorses for the cell. So first, what is DNA? So DNA is, as you've probably heard, right, deoxyribonucleic acid. That's what it stands for, DNA. And this is a molecule formed from nucleic acids that form the nucleic acids consist of phosphate groups, sugars, the deoxyribose, and nitrogenous bases. And so as we go along, we're going to talk a bit more about the makeup of DNA and how why it has its double helix structure. So there are 4 nitrogenous bases in DNA. And so as you've probably heard in your class, is adenine, thymine, guanine, and cytosine. And so we're also going to talk about how these pair together in a special way. So where is DNA? Where is it located? And what cell types have DNA? And so it's important to know that all living things have DNA. DNA is the blueprint for life. And so even single cells like prokaryotic cells, as we probably talked about bacteria, even up to multicellular organisms such as eukaryotic cells, such as you, right? Or make trillions of cells. And so where is this DNA housed? And so the DNA is located in the nucleus of the cell. And in prokaryotes, it's a bit different. We don't have these compartments, but we do have DNA that is free in the cytoplasm. Whereas in the eukaryotic cell, it is more housed, and there's more regulation that occurs in a eukaryotic cell that makes it much more complex. And so eukaryotic cells have DNA, as we've talked about. And so protozoas, plants, and animals are eukaryotes that all have DNA. And prokaryotes, such as bacteria, have circular DNA. And so it is a bit different, but it's important to understand that all living things, all living organisms, and bacteria are living, have DNA. And so what does this DNA look like? What's the structure, and why is it important? And so DNA is shaped like a twisted ladder. So you've probably seen in your textbook this helix shape, this double helix shape, and you've seen these bases in between these rungs of the ladder, if you will. These are the nitrogenous bases we spoke about. And so in this DNA structure, hydrogen bonds between the nitrogen bases from the rungs of the ladder form the rungs of the ladder, while the sugar and phosphate groups form the sides. And so the adenine and thymine base pairs and the guanine and cytosine base pairs are the stairs, right? They're what you're walking on in a ladder. And so it's important to note that here the adenine and thymine base pair together, and the cytosine and guanine base pair together. And so they have special properties that allow them to base pair with these particular bases. And so it's important in DNA that you understand that adenine and thymine base pair and cytosine and guanine base pair. And if you've probably heard about RNA is a little different. There's another base that's used instead of thymine, but we won't go into that depth at this point. And so there are the sugar and phosphate groups. They like water, and as we say, they're hydrophilic. So anything that's water loving or likes water is deemed hydrophilic. And the nitrogenous bases are not hydrophilic. They don't like water. And so what we call this is that we say this is hydrophobic. And here in the schematic, we see a phosphate group, which our phosphate groups have a negative charge. So DNA is negatively charged, right? And then we have our sugar, which is our deoxyribose, and then we have our bases. And in our bases, you see here in the schematic where it's a schematic showing inside and outside of the cell, demonstrating that the hydrophilic things like water and the nitrogenous bases are hydrophobic inside depicting inside. And so these physical properties of the nucleic acids cause DNA to have its helical shape. And so it's these characteristics and these physical characteristics that give the DNA this helical shape that you're so used to seeing in textbooks. And so importantly, so what does DNA do? And so we see this drawing or this blueprint here of a house, if you will. And it's really nice to think about DNA as a blueprint. And so we a lot of times think about connecting or comparing DNA to blueprints of houses, because the DNA serves as a blueprint for all of the information that is expressed in an organism. So like the blueprint for the house, the DNA in all of your cells contains all the information needed to build and maintain an organism. And it's important to note that every cell in your body has the same exact DNA blueprint. It is the regulation of this DNA, this genetic information, that allows you to have the diversity of cells that you need and allows for us to make all of these different proteins and all of these other molecules that are essential for maintaining life. So this includes the information for the machinery that will do the building as well as materials. And so you all probably have talked about ribosomes and RNA and all of these molecules that are very, very important to facilitating the process of going from DNA to RNA to proteins. And so the information housed within your DNA is important for providing the instructions to build these materials. And so DNA remains dynamic through the lifetime of the cell, making sure the cell has everything it needs to stay healthy. And so this is where we talk about the regulation of DNA. DNA is not a static molecule. You want to think about it as being very dynamic. It is being utilized to make different proteins or make things that we need that we're running low on. And so think about DNA as this very, very dynamic molecule that is working throughout the lifetime of the cell. And so all of the information that is in the genetic code is housed there, and we can use this genetic code to actually think about, well, what does each of these bases actually make? What amino acid, what code, what signal, what instruction is being given from this genetic code to make what we need to sustain life? And so now to think about this genetic code a little bit more, we're going to talk about the sequence or the order of bases and how important that is in the strand of DNA that makes the code and eventually will build protein. So here in the schematic, we start at the very top where we see DNA, and DNA is actually being unwound to be read and then also transcribed. And so after DNA is transcribed and then translated to make proteins, we can actually utilize this code to figure out what exact amino acids are being translated from this initial genetic code. And so the genetic code is a triplet. So we think about each of these triplets, such as ATG, CTA, and GGC in the schematic as codons. And so they are in 3 bases and they are called codons, and they can code for an amino acid. And some of these amino acids have several codes, but here we find that this ATG, which is a coding for methionine, is going to be the start of this process, and it's going to tell our friend the ribosome when to latch on and start this process of translation. And so I really, really like this analogy of thinking about a gene in the entire sequence of the bases that code for the specific protein as a cookbook. So here we have Patti LaBelle's cookbook, probably a holiday cookbook. And you could think about the genetic code or the blueprint for DNA as being a cookbook. So in Patti LaBelle's cookbook, you have all of the recipes that are important to making a Christmas dinner, right? And so you can think about DNA in the same way. Your DNA houses all of the information that is needed to make anything that you need in your cell to stay healthy. However, each specific instruction in Patti LaBelle's Christmas book will make a specific dish. And so we have specific genes that will make specific proteins. And so you can think about our DNA as the cookbook And we don't talk about RNA much in this talk because we're extracting DNA from strawberries, but we want to think about each individual recipe in Patti LaBelle's book as the RNA. So the DNA is made, RNA is going to serve as the intermediate that sends the instructions for a protein to be made. And each sheet in Patti LaBelle's cookbook can serve as an instruction for a specific dish, just like our RNA molecules are going to serve as this messenger for making a specific molecule. And this diagram seems a bit busy, and it is, and so you can just relax. We're not going to go too far in depth in this, but we have deciphered the genetic code. So what are these codons coding for, and what amino acids are coming from these codons? And so here in this wheel here, we have created a table, or in this schematic, a spin wheel, where we can actually look at the codon sequence and see what amino acids each codon, that triplet code that we just talked about, what amino acid is actually coming from this triplet codon. And so here we have A, T, G, which creates methionine. And so there's a very nice way to decipher this code here to the codon table to figure out what amino acids are coming from each of these triplet codes, which we do call a codon. And so importantly, the foundation of biology, molecular biology, is this process of the central dogma. The central dogma, you may have heard of, is the process of how we express our genes and make proteins and molecules that are very, very important to sustaining life and keeping our cells healthy. And so in this process, we have DNA, RNA, and an eventual protein. And so going from DNA to RNA, we are going to go through a process called transcription. And so we have evolved a process to express our genes through the central dogma. And because DNA is so important and it maintains life and is the blueprint of life, we want to protect the DNA in the nucleus. We don't want the DNA traveling outside the nucleus. We want to keep it housed in a specific place. So we've evolved a process to where DNA is actually transcribed and a message is sent from that DNA molecule to the cytoplasm via an RNA molecule that is eventually going to be translated into protein. And this is a very, very important concept, and it serves as the foundation, as I mentioned, for molecular biology, biochemistry, genetics, really for all of biology. It is a central concept of life and how we actually express our genetic information in cells. So I wish this could be more interactive to answer questions, but hopefully Ms. Owens can answer questions that you have, and we'll keep moving. And so to talk a bit more about how our DNA or our genes are expressed and how it shapes who we are, our DNA encodes the genes we inherited from our parents. And so in sexual reproduction, offspring gets half of their genetic information from each parent. So you get half from mom and dad. And these traits are passed on through genes from parents to offspring. And the traits are distinguishing features or characteristics. So it's important to note, why don't you look exactly like your brother or sister, right? And so in this very complicated process, in our sexual reproduction, our genes actually segregate, recombine. And so even though you're still getting genetic information from mom and dad, and if you have the same mom and same dad, you still don't look identical to your brother or sister unless you're an identical twin. We have this process that we study in genetics where you have recombination that allows the genes to assort in a different way to give different traits. So you can have curly hair, but your sister or brother may have straight hair. etc. And you may have blue eyes, but your sister or brother may have brown eyes. And so all of these traits that are passed down from your parents recombine in a way that allow you to be your own specific genetic code. So no one else out there, unless you have an identical twin, has your identical information as in the genetic code. And so now getting to the experiment for today, how can we isolate this DNA? And so first, we can isolate the DNA by breaking open the cells. So we are going to break open the cells today, but we are going to put methods in place that actually protect the DNA. So we're going to use detergents and salts to help us do this and help us break open the cells, but also protect the DNA at the same time. So first, we're going to crush the fruit to better help the detergent and salt free as much DNA as possible. And then we're going to remove proteins and cellular debris with the filter. And this is probably going to be your coffee filter that you're going to be using in class. And so remember that sugar and phosphate groups of DNA are hydrophilic. And so that's important to understand. And so the next step of the DNA is to precipitate the DNA from this strained solution using alcohol. And so DNA is not soluble in salt and alcohol. So we're going to preserve our DNA using this method. And it's important to know that alcohol is not soluble in the salt and the alcohol. And so now what do you need? So we have our fruit, we have our liquid detergent, we have our table salt, we have our warm water, about 100 milliliters of warm water, we have our ice cold alcohol, and then we have our sieve or our coffee filter. And then we also have something to remove the DNA, something like a Q-tip or some type of wooden kind of toothpick, if you will, to remove the DNA. And then also we have our personal protective equipment. So everybody should be wearing our mask anyway, but you also should have gloves and if needed, some eye protection. All right, guys. So I hope that you all have a great experiment this morning.